Good morning. Thank you for coming this morning. I'm going to tell you about a company called Celsense that was founded in 2005. Celsense is a company that makes tools that are used by people developing cell therapies, sort of the picks and shovels of the gold rush that we're hearing about this week. And I'll talk mostly today about the science, the technology, and I'll talk a little bit about the company at the end, the business. So the unmet need that Celsense fills is we sell and develop quantitative in vivo cell tracking tools. So these are imaging-based tools for tracking cells in the body. And this is, of course, important for cell therapy, we believe. The ability to track cells after you put them into the body is, I think, a common need of many of the developers, both for scientific reasons, but also to help overcome regulatory hurdles where bodies like the FDA would like to know where the cells go after you put them in. A related problem is inflammation, where you have anomalous influx of lymphocytes in tissues and accumulation in tissues, and so it's a related problem. So we have technologies that address both these needs. The products that we supply are imaging tools for discovery, preclinical, translational cell therapy, and diagnostic medicine. We currently sell into the first two markets. And so what's unique about our products is that you can take the same technology, imaging technology, and use it in a mouse, larger species, rats, monkeys, but also then bring it into man. So a single imaging assay can be used across the whole pipeline for cell therapies. We sell reagents, and there's three families of reagents that we sell. Celsense, Vsense, which I'll talk about in some detail, and a little bit about GeneSense. Celsense is a reagent that's used to label cells in culture, ex vivo, and then when they're transplanted into the patient, they can be tracked using MRI. Vsense is injected intravenously into a patient, and then it highlights sites of inflammation, which is they're detected using MRI. GeneSense is an MRI-based gene reporter. And I'll go through systematically through each of these technologies. We also have a software product called Voxel Tracker that maximizes the richness of the data set that you get out from Celsense and Vsense. The Celsense platform starts with cells of interest, and they can be any type of cell. They could be a stem cell or immune cell or any sort of progenitor cell. They could be autologous, allogenic, or an engineered cell line. Generally in cell therapy, you do a tissue culture steps or to expand the cells, modify them some way. Celsense is a product that's added directly into the culture media. It's a perfluorocarbon emulsion that's been engineered to readily enter into cells, thereby labeling the cells. It stays with the cell as long as it remains viable. The cell dies, it's released. The cell divides, it takes half of the dose with each of the daughter cells. It's very non-toxic, and there's no evidence for exocytosis, et cetera. Once you've labeled the cell, you can determine how much of the agent has been taken up by the cell. And then you put the cells into the patient and you do an MRI scan. You take a proton scan, which is the conventional anatomy that you're accustomed to, but then you take a fluorine-19 MRI scan. The fluorine-19 is what you detect from the perfluorocarbon. And so it's a unique signature, has no background signal, enables you to do overlay images, which allow it to look at the labeled cells, the anatomical context. And our software allows us to then actually count how many cells are present in regions of interest. So this is a technology I call in vivo cytometry, or counting cells in vivo. Now, another powerful way to use the technology is using high-resolution NMR. This is done in preclinical models where you inject the cells. You want to know the biodistribution of those cells. You want to determine that rapidly without having to do a lot of histology or flow cytometry. You can then biopsy tissue panels, place them into an NMR tube, and then use conventional NMR spectroscopy to generate histograms where the x-axis shows the tissue panel, the y-axis shows the cell number. This is what a labeled cell looks like. This is a dendritic cell. In this EM here, you see these little white dots here. Those are the perfluorocarbon droplets that are taken up inside of cell. Generally, they traffic into vesicles, low pH vesicles like lysosomal compartments or macropenosomes in the case of the dendritic cell. And they stay there. This is an example of the types of readouts that you can get with this technology. You can look at the injection site. So if you're delivering cells, you want to know if you hit the right spot. This shows the proton image of a mouse quadriceps. Asterisk shows you injection site. This is the fluorine signal. It's a fuzzy blob. You overlay the two data sets. You get the labeled cells in the anatomical context. You can see if you hit the right spot. You can also look at cell migration. Cells are injected in the foot pad, migrate up the ankle, the lymph node adjacent to the popliteal, into the popliteal lymph node. You can also look at the biodistribution of cells. So if you have no a priori knowledge about where they might go, you inject them IV, look for the hot spots of where they accumulate. In this case, they're weakly in the lungs, but also the liver and the spleen, sort of regions you might expect them to go to. And so the beauty of this, you don't need pre-scans to characterize your anatomy before and after injection. You take a single scanning session with the scanner, and you can look at the cell biodistribution. You can also look at stem cells. Here's an example published recently. This is a stroke model in a rat. This is the stroke region on one hemisegment of the brain. And the cells were then injected into the stroked region. And you can see that you actually made an accurate injection, and then you could follow those cells days and weeks later. Now, another powerful way to use this technology, as I said, is just using common NMR, which is most analytical chemistry laboratories have this. And you can look at cell biodistribution. So just necropsying tissues, putting them in NMR tubes, taking NMR spectra. You can quickly generate histograms that look like this. This is the tissues, x-axis. The y-axis shows, in this case, number of T cells in organ in this model of colorectal cancer IBD, this MUC1 IL-10 model. You see that there's lots of cells in the colon in this model. Unexpectedly, we saw cellular niches that were not known about, and they seem to home to the pancreas as well, suggesting these mice have pancreatitis. You could also do MRI of these same animals. This is the hot spot shown in the pancreas. This histogram allowed you to predict the result. And you generate this sort of NMR data just using a robotic sample changer on an NMR machine. It's very quick, so you can basically part out the mouse, run the parts overnight. Next day, you get the biodistribution of cells. This is a very important type of assay that you can do at the discovery and preclinical stages of cell therapy. We've also made dual mode versions of this reagent where we've conjugated a bright fluorescent dye, the perfluorocarbon, and you can then detect the cells in both MRI and then using fluorescence microscopy or flow cytometry, for example. And this is important for validation concerns. So you might inject a stem cell, excuse me, and you'll see some hot spot in your scan. You want to know, is that the stem cell or is that a macrophage that's come to eat a dying cell, for example? So with those validation concerns in mind, we've developed this technology. We've also conjugated it to NIR dyes, so you can do in vivo fluorescence imaging. And these are really nice, bright dyes with durability, and so we like them a lot. So you'll see more of this sort of technology in the future. Now, from the beginning, we've had an eye on clinical translation. And perfluorocarbons are actually very safe in the body. There's been a long history of putting perfluorocarbons in the body in the context of artificial blood substitutes. So the FDA is very familiar with the chemistry and pharmacokinetics of these materials. However, in our particular application, we're essentially putting in a de minimis amount of the material in the body. It's only contained within a labeled cell. So it's like microgram levels. In fact, we did a calculation that you ingest more fluorine into your body just by drinking fluorinated drinking water for 1 day compared to what you would get with one of these scans. And so we had discussions early on with the FDA. We devised a novel regulatory path for this material where we've opened up a drug master file for the Celsense. So drug master files historically treated as something you'd use for an excipient to a therapy, like a bulking or a colorization agent. In this case, we're actually using an MRI agent. We're going to the FDA through a drug master file. So this is open. It contains the CRC data, acute tox data, cell tox data, genotox data, et cetera, manufacturing data. And so this is available. And so the investigator would hold their own IND. They cross-reference the drug master file when they send it to the FDA. So in this way, we've enabled the ability to disseminate this technology to a large number of different types of clinical trials. And I think this model might work pretty well. So I think it's the first example of this. It's a novel regulatory path. The first IND that we filed was a phase 1/2 clinical trial at the University of Pittsburgh Cancer Institute to treat stage 4 colorectal cancer. The investigators there have developed an autologous dendritic cell vaccine. And we're comparing different routes of administration to see what the efficiency is, migration to the lymph nodes, which is an important information the clinicians would want, but there's very few ways to actually get that sort of data except for, I think, our technology. And this was funded by the NCI. We've recruited our first patient in early this year, And the technology seems to be working well. Just in a conventional 3T clinical scanner, we can see the injection site. In this case, it's an intradermal injection. And then we can watch that injection over time. So this is some of the first clinical data that we've gotten with this material. And there's many other INDs that people are preparing that intend to use this type of technology. We've labeled many different cell types of interest, including immune cell types, dendritic cells, T cells, NK cells, B cells, many types of stem cells, and neuroprogenitors and things of that nature. And so it looks like this is a very versatile technology. Now, inflammation. So we've formulated perfluorocarbon emulsions for a very different purpose, for injecting intravenously, and then these emulsion droplets are taken up by the RES system, including monocytes and macrophage, which then participate in inflammatory events in the body, thereby accumulating fluorine, showing hotspots. And so you just inject the material, you wait about 24 hours for the material to enter into the RES and then participate in the inflammatory events. And then you take a fluorine-19 scan, a proton scan, you overlay the two data sets, and you get the hotspot of inflammation in the anatomical context. Now, in this case, we can't actually count cells because we don't know how much material is taken up per monocytes, but we can calculate the total amount of fluorine accumulated at that hotspot, and that's linearly proportional to the macrophage burden, and we've shown that using molecular means. Another powerful way to use this technology is the NMR, where you necropsy a panel of tissues, drop them in an NMR tube, no other processing. NMR spectra, you generate these histograms. Tissue on the x-axis, something we call the inflammation index on the y-axis, which have units of fluorine atoms per gram of tissue. And this, again, reflects the macrophage burden of that tissue. We're assaying this without having to cut the tissue, without having to disassociate it in a single-cell suspension. So it's very rapid. Here's some examples of very versatile technology. I'll show you a couple of nice examples. Like this is organ transplant rejection. Heart lung from one strain of rat is grafted into a different strain of rat, such that it undergoes rejection. You do a single then IV injection of Celsense, and then you image 24 hours later, you get something that looks like this. This is a transplanted heart that's actually grafted into the organ, into the abdomen, into the blood supply, and you see a ring of fire around the periphery of the transplanted heart. This indicates the macrophage pattern. So if you integrate up the heat associated with this, our hypothesis is that you can stage the rejection. The common clinical practice to take serial biopsies, and those have a lot of drawbacks, as you know. And so this is a way to non-invasively assay organ transplant rejection. This panel here just shows a high-resolution MRI of the heart that's been removed. Another example, inflammatory bowel disease. Something that's often hard to diagnose is a patchy disease. In a conventional MRI, you can often see a thickening of the colon wall, but there's some subjectivity to examining those scans. With Celsense, there's no subjectivity. This is ring of fires around the colon, the ascending, descending colon. And you see that the heat of this represents the severity of the inflammation. And then if you have a therapeutic test article that you'd like to test in this model, you can then see whether that heat is diminished, whether it's working or not. This is a 3D scan of the colon acquired in vivo, ascending, descending colon. Down here is the anus. This is acquired in about 8 minutes of scan time on a preclinical scanner. We also have dual mode versions of the Celsense that have an NIR dye conjugated to it, so you can do fluorescence in vivo imaging. And this is in a tumor-bearing animal, a mouse, and this is the liver. Here is the tumor here. It's the control, no tumor. It's the liver. It's taken up by the RES, so that always lights up. And this is what the same animal looks like in MRI. You can see nice definition of the tumor. But moreover, you get 3D examination of the tumor, and you can measure its volume and so forth. And so it's very effective at looking at the tumor-associated macrophage with high resolution. Now, we've also developed a software product called Voxel Tracker, and this does the overlay of the proton fluorine and the pseudocoloring, but it more importantly has quantitative tools that are built in to assay the number of cells, to calculate the number of cells by drawing regions of interest, and then calculating the apparent cell number. It gives error bars for the result. I'll skip Celsense for the sake of time. I'll just say a word or two about the company Celsense. We believe that we're thought leaders in cellular molecular MRI. We were founded in 2005, so quite a while ago, It's a spin-off of Carnegie Mellon University, my old employer. We have manufacturing laboratory offices in Pittsburgh, Pennsylvania. We have a very strong IP portfolio. We've been very aggressive about patenting this since the beginning. And we have a small company, three full-time employees, and we finance via angels and grants. $10 million are raised. And the ask today for you all is we're looking for customers. We're looking for strategic partnerships. Thank you.